VGAS, generating coverage consensus views. The coverage button located on the bottom right allows the user to display and analyze coverage and consensus information. The coverage view can either be generated genome-wide or for the currently selected gene or feature. The coverage option form includes a sequence grouping option panel, which allow us to create different coverage groups if needed and also place cutoffs. If the none option is selected, then all sequences will be used to create a single coverage group. Selecting sequences allows us to create as many groups as we like, with each containing the sequences we select. The plus sign creates a group containing the selected sequences. So, select sequences by either using the mouse or the Shift or Control key. The minus sign either removes an entire group or a sequence from the group. The group name can be edited by clicking on the group name or root. If the sequences have been loaded from a FASTA file or Sanger data, then each sequence usually represents a single sample. However, if they have been loaded from a SAM or BAM file, then it's most likely that next-gen data, and each sequence or row represents a number of reads, and each single sample may cover quite a few rows. VGAS will list each row for each sample in order, showing the identifier followed by an underscore and the sample row number. Identifier underscore row number. If the next gen option is selected, then the sequences will be grouped by the identifier or sample, i.e. the identifier up to the first underscore. The number in the brackets being the total number of rows. A cutoff can be set to discard any nucleotide bases within a group which do not meet a value at a single position. This value can be set as a base count, percentage, or read percentage. For example, if you enter 20 and select base count, then if there's only 19 base A's at position 100, then these will not be included in the coverage. The percentage option is more suited to FASTA or Sanger. The entered value is used at each nucleotide position and includes any unsequenced areas, shown as asterisks. The read percentage option is more suited to next gen. The value uses the read depth at each nucleotide position and ignores all rows where no read is found, i.e. there's an asterisk. The update button will generate and display the coverage view. The message, update completed, will let you know when it is finished. Another thing to note is that the options form doesn't need to be closed when generating views between different genes or features, or genome-wide. So you can set up your grouping and then select other genes. Hit the update button and the coverage will be generated for that particular gene. So, a few tips regarding memory usage when generating coverage views. Obviously, larger genome-wide coverage views will require more memory than just a gene or a feature. So the greater the base count, the greater the memory needed. When generating the coverage, both the nucleotide and amino acid differences are worked out between all the groups. So number of groups added will also require more memory. The easiest way to monitor the memory while the coverages are being generated is from the Performance tab in the Windows Task Manager. Here you can see if you have sufficient memory to complete the task. However, if the memory maxes out, this doesn't always mean that the task won't complete. VGAS will keep trying to clear some memory. However, if it is taking a while, then you may need a computer with more RAM. Or VGAS also includes a coverage viewer, which can be opened separately in a different window. This option allows users to generate coverage views which take up far less memory. This will be covered in the tutorial video, the coverage viewer. Our next video will cover visualizing the coverage view.